Chapter 15 is not so far off from what we've been talking about, membranes and transport across membranes, because it's talking about protein sorting. So we've talked about the composition of membranes having a large component being proteins, these transport proteins, whether they're transporters or channels. But there are a lot of other proteins in the cell, even in the cell membrane, that have other functions. And when we're talking about eukaryotic cells, we're talking about a lot of compartments within the cell, right? Those organelles within the cell. So in order to get the protein to the right place to do its job is really critical for the function of the cell, right? If the protein, after all those steps, it got through the quality control steps of having a mature transcript, the transcript was then moved out of the nucleus where it could be translated into a protein, but then the protein doesn't get to the right place in the cell. That's a lot of energy that the cell has expended for pretty much no payback in the end, no function of that protein in the end. So you've got to get the protein to the right place in the cell. And there are many mechanisms that ensure, many steps along the way that ensure that, one, the protein is made, it's folded properly, it's functional, and that it gets to its proper site within the cell. But basically, one of the most important parts of getting the protein to the right site is going to be part of its sequence that in some cases is removed after it gets to the proper site in the cell, called a signal sequence. Okay, so let's just review for a minute. Okay, we know, we've been talking about eukaryotic cells. We know they have lots of organelles. A few of them are listed here. What other organelles are going to be found in a cell? So we have the main compartment of the nucleus. Nucleus, what's the function? Okay, it's going to carry the DNA, that genetic information. Then we have the endoplasmic reticulum, either smooth or rough. What happens in the rough endoplasmic reticulum? Protein synthesis, one of the sites. What is not labeled here? That would be another important organelle in the pathway of a secretory protein. Okay, the next compartment, it would move from where it's synthesized in the rough ER to the Golgi. And then it may go to the plasma membrane, lysosome, peroxisome. In the case of mitochondria, are those two membrane compartments, what do we call them to describe their function? Not symbiosis. What is their function? So respiration, we call them the powerhouse of the cell because it's making energy, right? Some of the proteins that are important for function within the membranes, either the outer or the inner membrane of mitochondria, are actually synthesized in the cytosol and then get transported directly across those membranes. So there are a variety of ways that, or three major ways that proteins can get moved around from where they're made, either the cytosol or the rough ER in the cell. And there are three major sites that have different functions. One is the nucleus, so it has that two-layer membrane of the nuclear membrane that has these holes in it that are the nuclear pores that allow for, selectively allow for things, molecules to get in or out of the nucleus. Then directly across the membrane in the case of mitochondria, or through vesicles in the case of moving from an ER to a Golgi, or a Golgi to the surface, or to be secreted outside of the cell. And I've just put this table in just to remind you that you should review the organelles in a eukaryotic cell, and you should know their functions. But basically, with all these compartments, the cell has a task to do in making sure that the proteins that it makes or synthesizes get to the right site. And what really helps are the signal sequences, which you can think of as a short sequence of now amino acids, okay, at the end of a protein that are recognized by chaperone molecule proteins or channels that help them to get across membranes or into the nucleus or out of the nucleus. One of the most important pieces of information that a protein will have that will determine where it goes in the cell is a signal sequence. Now, signal sequences are not found on all proteins. In fact, the function of a signal sequence was understood when it was genetically engineered. A protein that had a signal sequence was genetically engineered to lack that signal sequence. And it was an ER protein. And what happened was, by expressing the protein without the signal sequence, it ended up staying in the cytosol. So we know that cytosolic proteins lack a signal sequence. So as soon as that message comes out of the nucleus is in the cytosol, it's going to be translated into a protein, whether it's destined for a compartment in the cell or destined to stay in the cytosol. But the major difference between those two groups of proteins is that cytosolic proteins don't have a signal sequence. And the other proteins that are made either on the ER or even in the cytosol do have a signal sequence. So they will not remain in the cytosol. So signal sequences, I don't expect you to memorize these different signal sequences, but they are composed of amino acids because they're part of the protein, so they're part of the coded information in the messenger RNA. That results in a sequence either on the N-terminal end, so the very first part that comes out of the ribosome. And here, the ones that are N-terminal have the amino group indicated. Can be at the C-terminal end as well, or somewhere in the middle. And while the specific sequence can sometimes be important, so the serine, lysine, leucine sequence indicates just 3 amino acids indicate that that protein needs to be transported to the peroxisome in the cell. It's not always the actual sequence, but it can be simply the properties of those side chains that the amino acids have that also contribute to whether the cell contributes to the information that helps the cell get that protein to the right location. Okay, so to get into the ER, has to be at the N-terminus end, because what happens, know about are destined for the ER, where are they translated? So the ER, there are ribosomes studded along the rough ER, and that first step, translation, after the translation is initiated, by the N-terminal end having that signal sequence, the ribosome is then moved with the help of proteins that bind both the ribosome and the protein that's in the process of being synthesized to the ER membrane. So it doesn't start where the ribosome attaches to the ER membrane. Instead, depending on what message starts making the protein, some of the ribosomes will remain in the cytosol, some of them will move to the ER membrane and be those that compose the rough ER. So those proteins that get into the ER, they may remain in the ER, in which case they have to have another signal sequence at the other end of the protein. That will indicate that they won't be shipped anywhere else in the cell. Some of those proteins will then, if they lack that retention after they fully fold, and possibly get glycosylated in the ER, will then move to the Golgi and by via vesicle mediated transport, which is one of the mechanisms that we'll talk about today, and then may remain in the ER or may move on to another site in the cell, either the plasma membrane, they may be secreted outside of the cell, or they may come back in from the plasma membrane into the endosomes, which then mature into lysosomes. There are a couple of other ways that proteins get shipped around in the cell, but they also are made in ribosomes in the cytosol. An N-terminal signal indicating import into the mitochondria is important for the shipment of that protein after it's made to get to across the mitochondrial membrane. So the ribosome doesn't attach as the protein is being synthesized, okay, as that translation is occurring. Instead, after the protein is completed, it doesn't remain in the cytosol. It moves to the mitochondria. And that's direct transport across the membrane. And then in the case of the nucleus, most of the membrane-bound organelles have a simple bilayer that separates them from the rest of the cell. But in the case of the nucleus, remember this is a two-fold membrane that's kind of folded in upon itself that forms the barrier between the nucleus and the rest of the cell, but also forms the ER. Okay, so it's kind of the lumen of that space between those two membranes is actually the lumen of the ER, and it expands as the cell needs. If it's very busy making a lot of ER proteins, that ER can expand or it can decrease as the cell needs. So export or import into the nucleus goes through special openings between these two bilayers that form the nuclear membrane. And they're called nuclear pore complexes. And so it's a little bit different mechanism through the that is involved in import to the nucleus. Again, the proteins are made in the cytosol. Once they're fully translated and folded, then a protein receptor will bind and recognize the signal sequence and help transport it through the nuclear pore. And some proteins have a similar sequence that helps them get the other way across the nuclear pore. So basically the charge, whether it's a positive charge with lysines and arginines, or a negative charge for the retention and the lumen of the ER as far as glutamine and asparagine, those will have an influence, as well as the hydrophobicity. So remember, lipid bilayers are hydrophobic on that inner region where the tails are. So sometimes it's helpful, especially if it's a transmembrane protein, to have regions of hydrophobicity to get across that membrane into the compartment. Okay, so experiments have been done, as I mentioned, to understand the function of these signal sequences, the ER sequence being at the N-terminal end of a protein, and by genetically engineering these proteins to lack a signal sequence, or even by putting the signal sequence found on an ER protein and genetically engineering it onto a cytosolic protein, we can control where the proteins are shipped in the cell. So this blue ER protein, if you remove the signal sequence, becomes a cytosolic protein. And so these signal sequences are really key in the shipping process within the cell. If we add a signal sequence to a cytosolic protein, here indicated in red on the green cytosolic protein, it will no longer be a cytosolic protein, it will be an ER protein. Okay, so proteins are either made in the cytosol, as I mentioned, or started in the cytosol but finished in the rough ER space. And these are the proteins that are part of the endomembrane system. So remember, the endomembrane system is a subset of all the membrane-bound organelles. And this subset is grouped together because they tend to have a common pathway. So proteins that are made in the ER go to the Golgi if they're not ER proteins. So the ER, the Golgi, the peroxisomes, endosomes, those are all part of the endomembrane system. So ER, Golgi, endosomes, peroxisomes. So not the nucleus, it's not associated with that subset of organelles, and not the mitochondria. So we already went over translation, we went over the function of the mRNA being that it holds that triplet code that tells what tRNA to bring, the tRNA to bring in a certain amino acid, and so that polypeptide chain, the fidelity of that polypeptide chain is ensured by this codon-anticodon matching. But what happens in the shipping process after proteins are synthesized, and what happens to proteins that are destined for the ER, for the endomembrane system? So as I mentioned, shortly after the beginning of translation occurs in the cytosol, the ER signal sequence will indicate to the ribosome to attach to that rough ER. And it can happen just like in the cytosol. You can have more than one ribosome attached to a single transcript, right? And same thing can happen on the ER. So there are a lot of these translocator channels through which a new protein can be shipped as it's being translated. So while there's this common pool of ribosomes in the cytosol, some of them are used on the rough ER, and then once that translation is complete, the ribosomes disassemble and remain as part of that pool in the cytosol. After that translation is complete, the polypeptide, or the complete protein, remains attached to the ER for a period of time until other information in the protein is read, or other interactions occur that help to properly fold as well as properly locate the protein. So in the case of an ER luminal protein, it will pass all the way through this this transport channel into the lumen of the ER. And if it has a signal sequence that it's an ER protein, it will remain in the lumen, and the signal sequence that located to the membrane of the ER will be cleaved so that it will become a soluble protein in the lumen of the ER. So chaperone proteins that help other proteins to fold properly are an example of a protein that would be shipped in this way. But as I mentioned, there are three major ways. So other than across membranes, such as the ER or proteins that are made in the cytosol, can cross the mitochondria, or even in plant cells, the chloroplast, which is analogous to the mitochondrion. But they're also, for proteins that get into the lumen of the ER, whether they're transmembrane proteins destined for the plasma membrane, or whether they're lysosomal proteins that have to get into that compartment, they're going to be moved around by transport vesicles. And then proteins that act in the nucleus are going to be transported via a third mechanism that involves the shipping across through the nuclear pore. So who can give me an example, thinking back to some topics that we recently talked about, of a protein that would need to be shipped to the nucleus? We spent almost a whole chapter on this topic. Control of transcription occurs thanks to transcription regulators, right? So all those proteins, all those repressors, all those activators that are in eukaryotic cells, those transcription factors that we learned about, They need to get to the nucleus. I mean, there's no way that they could have any effect on what DNA is being transcribed if they're sitting back in the cytosol, right? So those kinds of proteins have a nuclear localization signal that helps them get through those pores in the nuclear membrane. So the first step is the making of the protein. And we already went over that, so I'm not going to go over that in detail. So the protein, the mRNA is translated to make a protein. Then here's an image of an electron micrograph, kind of a slice through the nucleus, so looking on from the side here. This is looking kind of from the outside in. And these are actually, these are not in an intact cell. These are pores, nuclear pores that have been isolated from the membrane. So they're all kind of packed in together here. But here you can see that they are oriented and spaced kind of, with some distance between those two lipid bilayers of the nuclear membrane. And this is, here is a nuclear pore, and it has, you can see a little bit of the basket shape of the part of the protein that hangs off of the complex of proteins. So it's actually not just one protein. The nuclear pore is a complex of about 30 proteins that make up this very symmetrical looking pore. The inside that nuclear pore, there's kind of a gel-like matrix that's formed by a meshwork of unstructured regions of the proteins that make up the complex that kind of hang out into the center of that pore and basically inhibit anything from freely flowing in or out of the nucleus. But thanks to some specific interactions and proteins, other proteins, factors, proteins that are active in the nucleus, like transcription factors, can get through the pore. So here's the cartoon structure. There's actually a lot more detail given in the current version of the book, in Chapter 15 in the figure. But it has a basic shape of these 30 or so proteins assembling together to form the pore, and then some fibrils that hang off on either side. This is the nuclear side, and this is the cytosolic side, that help also with attracting proteins that need to be translocated. And there's little baskets, so these fibrils are then form a kind of cage-like structure that you saw in the electron micrograph in the last slide, because they have a ring, a protein that forms a ring at the end of those fibrils. So they're actually connected, not just free-floating like the cytosolic side is. And along that inner membrane of the nuclear envelope, there's a matrix of proteins called the nuclear lamina that helps to kind of hold a bunch of proteins as well as nuclear components in place. So this is really how everything, except for those small particles that are not charged, get in and out of the cell, in and out of the nucleus. They're the gatekeepers. It's a little different than the selective permeability that we talked about with the plasma membrane. And it is selective in, this pore complex is selective in what can get across into the nucleus. And the nuclear localization signal in a protein, represented by the red dot here, will be recognized by a nuclear receptor, a nuclear transport receptor protein, another protein, that will then bind and form a complex with that protein that needs to be shipped to the nucleus and help actually get it through this gel-like matrix that plugs the nuclear pore complex or the nuclear pore by almost like a slinky going down the steps, kind of stepwise interacting with a very specific but repeated sequence of amino acids that are on the unstructured regions of these proteins that kind of stick out into that opening. So it'll go kind of step by step through. It's not a slow process, very quick, happens in a matter of milliseconds or seconds. But it's definitely a stepwise process where the protein will not be able to get across unless first it binds to its receptor protein. That receptor protein then interacts with the proteins in the pore complex. And then once the protein is inside the nucleus, the dissociation of the receptor and the nuclear protein occurs. So how does the receptor know when to bind? Why doesn't it just go through the pore empty? And how does it know that it's on the other side, the nuclear side, and it's time to release that nuclear protein from the interactions that have facilitated its shipping to the nucleus? Well, as with all of these three mechanisms of transport, transport across and shipping into the nucleus requires energy. And it's GTP binding that catalyzes the dissociation event here once the nuclear protein is in the inside the nucleus, and then that receptor is then shipped back out empty. And then it's the loss of the GTP that kind of recycles it for another binding with another nuclear protein. Okay, so these are energy dependent. These are active transport mechanisms. They all require energy in order to occur. And in this case, it's in the form of GTP. So there is also transport in the other direction, not just for the receptors that are helping to transport proteins in, but also for other molecules in the nucleus that need to get out of the nucleus. So we already talked about one important type of molecule that needs to get out of the nucleus when we talked about gene expression. And what is that molecule? mRNA, right? So any type of RNA, even the RNA that's involved in the ribosome. So once it's assembled in the subunits of the ribosome are assembled, they need to get out of the nucleus in order to function in the cytosol. So the transport through the pore occurs in both directions. But in all cases, it involves proteins. So the mRNA itself does not have a signal sequence. Instead, remember there were some 3 steps that had to occur to go from the pre-mRNA to the mature mRNA. And proteins associate with the poly A tail, the 5' cap, the merge sites between exons where introns have been removed. So those proteins are the ones that are interacting with other types of transport receptors that will help them to move outside of the nucleus. Okay, so that's transport across the nuclear envelope through nuclear pores. There's also transport across membranes. And I already talked about ER luminal proteins, but proteins need to get to other sites as well. And that includes the mitochondria. So proteins that belong in the mitochondria also have signal sequences. So they'll be synthesized so the translation event will occur in the cytosol, but they won't remain in the cytosol after they're folded because of their signal sequences. And there are, again, receptor proteins that bind the signal sequence and move that protein to the site of a mitochondrion within the cell. So once that receptor protein is bound by the signal sequence of a mitochondrial protein, and there are transmembrane receptor proteins for some mitochondrial proteins. They will be closely associated with a transporter protein that will form a channel through either the outer or both the outer and the inner mitochondrial membranes, depending on the destination of that protein. So the signal sequence for the mitochondrion then, and remember that although the mitochondrion has its own DNA, it's actually most of the proteins in the mitochondrion come from those genes that are encoding proteins in the nucleus of the cell. So most of that machinery that's needed for this energy or ATP producing factory, all of that metabolism that's involved in catabolism in the cell, are actually encoded, most of them are encoded by the genome in the nucleus, not in the mitochondria itself. So the signal sequence is then passed from the receptor protein to the channel protein, and while the protein may have been fully folded, because it's fully translated in the cytosol, here it actually has to unfold. So there's not that additional step in the case of the ER proteins because they're actually being synthesized as they move through. So they'll fully fold in the lumen of the ER. But in the case of mitochondrial transport, the protein has to be shipped kind of in that primary structure as that string of beads. And then it will fold into the mature protein. And that signal sequence will be cleaved off in the process of maturation, and it will remain in the matrix. So again, this as well is requiring energy, an energy requiring transport process. Similarly, there is transport across membranes to the peroxisome, but in this case, the channel is large enough that the protein can move through fully folded. So that's one important difference between transport across the membrane of the peroxisome and the mitochondrion. And the chloroplast transport is similar to transport across the mitochondrion or mitochondrial membranes, where it has to unfold. Okay, so a third mechanism of transport is transport in tiny membrane-bound compartments that are vesicles. And this is the transport mechanism that is used to get to ship proteins that are involved in some compartment within the endomembrane system. So all of the proteins that are part of the compartments in the endomembrane system have to start out with translation on the rough ER. Once they are fully translated and translocated into the lumen of the ER, some proteins spend quite a bit of time there, because the ER is really the checkpoint at which proteins that are destined for the Golgi, the plasma membrane, the endosome, the lysosome, they are checked to make sure that they're properly folded and properly modified if they need sugars added or to be cleaved in some way. So the ER lumen has a lot of proteins that fall into a family of chaperone proteins, where they hold on to that protein until it's fully, properly folded and don't release it for transport by vesicles until it's assembled. This is critically important for proteins with quaternary structure, okay, so multiple subunits, disulfide bonding, those covalent bonds between the subunits of quaternary proteins occurs in the ER, and they won't be able to leave the ER until those modifications, whether they're covalent bonds between subunits or covalent attachment of sugars occurs, they'll remain held to those chaperone proteins. So it's really the quality control compartment. Nothing gets out unless it's really ready to go. Okay, so they get into the ER or into the endomembrane system by the ER sequence, and then rather than a signal sequence, there are other interactions, either specific or non-specific, that help them to get packaged into these tiny membrane-bound vesicles that then are used to move them from first the ER to the Golgi, and then the Golgi to other sites, either on the surface of the cell or inside the cell. So let's look in a little more detail at the requirements for that first step of how the ER protein gets across or gets fully translated. So one of the proteins that I mentioned but didn't name is the particle. This is the protein that binds both the signal peptide at the N terminus of these proteins that are made on the ER, and it binds the subunit of the ribosome. It doesn't bind that messenger mRNA, just the ribosome and the new polypeptide at the signal sequence. In that way, it helps to guide this whole complex, the intact ribosome, the messenger RNA, and the growing polypeptide chain to a signal to a receptor in the ER membrane that binds to another part of the signal recognition particle. And that receptor then brings the whole complex in close association with a translocation channel, passes off, so the signal recognition particle will release the signal sequence, which is then recognized by the interior hydrophobic or hydrophilic region of that translocation channel or that channel protein, which then holds the signal sequence through non-covalent interactions in place until the entire polypeptide chain, the whole process of translation, is finished. Then once the stop signal is reached, then the ribosome and the mRNA will just fall off. That complex will be broken. There might be other ribosomes at other points on the messenger RNA still holding that messenger RNA to the ER surface, but for that protein, it's done. The rest of it, in the case of those soluble proteins, will go through that translocation channel. And then if there are other signals, so that if there are no other signals, the signal sequence will be cleaved and it will become a luminal protein. But if there are other signal sequences in the middle of the protein, so remember I said there are different locations of signal sequences. In the case of transmembrane proteins, there needs to be signals that indicate that it's not going to the lumen, it's staying in that lipid bilayer. So for a simple transmembrane protein that only passes once through or across a membrane, no matter where it's going to be shipped in the cell, at the point that it's made on the ER, it has another signal sequence here indicated in yellow that means stop transfer. Okay, so that channel protein will then release the protein, whether it's finished or not. The ribosome will finish its job. Part of the protein will remain outside on the cytosolic side, and the N terminal will be on the ER lumen. Okay? And it will be released from that channel and just will remain in there's no channel in this cartoon. This is just the lipid bilayer of the ER membrane. Now, transmembrane proteins we talked about, some of them form channels, right? So they have many times that they pass through or across the lipid bilayer. And so they would have multiple stop and start transfer sequences. So depending on how many they have, they may not be released from the translocator channel or transfer channel until all of those sequences are read. I already mentioned that there's the first or N-terminal signal sequence is cleaved, and that's not by the translocator channel, but by another protein, an enzyme that will associate with that channel. And in the case of the luminal proteins or soluble proteins, it will cleave and release the entire mature protein into the lumen of the ER. In the case of transmembrane proteins, it just cleaves and releases that N-terminal end.
So is the structure then preserved in transfer vesicles until the cell wall in the cell membrane?
Right, so once it's folded, that structure won't be changed. It'll only be modified either by glycosylation or by covalent bonds as far as disulfide bonds and stuff. Once that finishes in the ER, then that's the structure it has, and if it doesn't do the job correctly, it doesn't get out of the ER. So the chaperone proteins will hold them in that space until they fold properly or until they get actually translocated back to the cytosol where they would be degraded if they'd never fold properly. So this is a really critical step in the quality control and ensuring that anything that gets through the endomembrane system is a proper functioning protein. So this can happen with transmembrane proteins that have 2 or 10 transmembrane, regions that go across the transmembrane, across the lipid bilayer. Just depends on how many of these start transfer sequences and the stop transfer sequences they have. And these are usually strings of amino acids that have some charge that they could interact well with the translocator channel, or they have hydrophobic properties that would allow them to remain in that lipid bilayer. So remember that transmembrane proteins have a common secondary structure. So when we learned about, in Chapter 4, when we learned about the different levels of protein organization, what are those 2 forms that the secondary structure has? Mohammed? Beta sheet and an alpha helix, right? So these regions that remain in the membrane would share those properties, that secondary structure. All right, so assuming that you have passed all of the quality control mechanisms that exist in the ER to make sure that you've got the protein, it's properly folded, it's assembled into a quaternary structure if necessary, it's glycosylated if necessary. There's a certain direction that the cell has in vesicle transport that generally goes from the ER to the Golgi, Golgi to the plasma membrane or extracellular space, or to the endosome, which can then mature into a lysosome. This direction is ensured because vesicles can transport items in both directions. Okay? So there's the cis side and the trans side of the Golgi stack of membranes. And just because of the proximity, the vesicles will butt off of the ER and fuse with the Golgi at the cis side. If any ER proteins, by mistake, get packaged into these vesicles, they simply get returned. Okay, so this kind of vesicle transport is continuously happening in both directions, even though the movement or the shipping of the proteins seems to be happening in one direction. And that just makes sense because you have to kind of get the bit of protein, the bit of bilayer that you pinched off from the ER back to the ER. Otherwise, you're going to have like this serious loss of the ER compartment as you're moving things around in the cell, right? And while it is dynamic and it does change with the needs of a cell. So if a cell differentiates from a B cell to B lymphocyte, a white blood cell that has an antibody receptor on its surface, to an antibody secreting cell, it's going to have a much larger ER. These antibodies have quaternary structure. They have the heavy and the light chains that need to be covalently bonded. They're bonded by disulfide bonds. They get modified, and if they aren't, and they get assembled together in that quaternary structure in the ER, but then they get constitutively shipped out of the cell. Their function is in our bloodstream, not as part of those plasma cells. So secretion happens through vesicle transport. And there's 2 general directions because there's secretion, and then which is also called exo or out of the cell, cytosis. And then there's also a process to bring things inside the cell by endocytosis. So phagosomes on the other kind of end of the spectrum, those neutrophils and the macrophage that are part of your white blood cells that are constantly like the sentinels of our immune system checking the environment for any kind of pathogen or foreign thing in its midst, it's constantly very busy doing endocytosis, right? So part of phagocytosis, that ability of it to engulf and destroy a foreign agent like a bacterium or a virus, is dependent on the endocytic pathway, and that's dependent on this vesicle transport. So how do these vesicles know what cargo they're going to carry, what proteins are going to be packaged into these little compartments, and how do they know where to go in the cell? Part of it is happening at the compartment where the packaging occurs. Part of the selectivity is happening at the compartment where they're destined. These kind of little cargo containers are destined to travel. And part of it depends also on motor proteins that are part of the cytoskeleton, because these vesicles travel along the cytoskeleton of the cell, which also kind of helps to orient the compartments within the cell. So at the site of the ER, for example, there would be a receptor in the membrane that would recognize that protein that needs to be secreted, for example, the antibody molecule, immunoglobulin. And it binds to part of that protein and accumulates. So here we only see one kind of receptor ligand interaction. It would actually be many that would accumulate in an area of the membrane of that donating compartment. And then once you get kind of a threshold level, the other proteins that are involved in helping that membrane to pinch off and form a vesicle, clathrin, represented here in green, form a matrix or kind of cage. So these proteins form a cage. They interact with the lipids and form a cage-like structure that helps to pinch the lipid bilayer off, along with key molecules that interact with their ligand on the donating compartment. So the V snares or vesicle snares are found kind of like the key that you find in the vesicle, and the lock are the T snares or target ligand that the V snares interact with. So once that vesicle finds, and so there are many V snares on any one vesicle and many T snares on any one compartment that it's destined to be shipped to. Once you get the interaction between a very specific interaction, because there are different types of V snares found on different vesicles and different types of T snares found on different compartments. So the plasma membrane T snare will not be the same as a T snare on the Golgi. And so you get a specific interaction that will hold the vesicle close enough to that new compartment to lipid bilayers will fuse. Once that fusion event occurs, then the contents in the lumen and the inside of that vesicle are released into the next compartment.
And like using bubbles, like putting your finger through.
Yep, it could be. So the actual interaction of these proteins causes an allosteric, so a shape change that you can actually see from crystallograph studies that they actually kind of twist, kind of like you would twist a corkscrew to get the cork to move out of a bottle. They twist the two membranes so that they're closer and closer together until they fuse. And this is an energy-dependent process. There are RAS proteins that are GTPases, actually, that use GTP to catalyze this fusion event, these transport vesicles fusing with the next membrane, target membrane. So again, there are specific steps in vesicle transport as well. First, the cargo has to be interacting specifically with a cargo receptor. Different cargo receptors will be in different vesicles. And then the vesicle, thanks to the clathrin coat, will pinch off. Also an energy-dependent step. And once it makes a specific interaction, so here's a different kind of V-snare represented by this kind of pink-colored with the pointy part of the shape, a different shape protein that interacts specifically with a different T-snare on a different compartment. And that docking is the interaction where the V-snare meets its cognate receptor or ligand, the T-snare. And then the membranes fuse and the cargo releases. So those four basic steps are common to all vesicle transport. So when I talked about antibodies, that was an example of secretion or exocytosis. And antibodies in a plasma cell are constitutively without any kind of regulation. As soon as that B cell matures to a plasma cell, as soon as those antibody molecules are made, they're going to be going through that transport process of ER to Golgi to the outside of the cell, secretion. But in other cases, the cargo in the vesicles stops at the plasma membrane and waits. In that case, we call it regulated secretion. So in a pancreatic beta cell, here in the electron micrograph here, you can see that this cell has been triggered to release its contents, which were waiting at the plasma membrane. This is outside of the cell. This is inside of the cell. Here's a complete vesicle. It's carrying a bunch of insulin. That's kind of stuck together there in the very dark part of the electron micrograph. And it's been triggered by signals through the plasma membrane of other receptors, so signaling that senses the environment that there's a need for more glucose, so the insulin needs to be released in order to transmit that signal that more glucose needs to be released into the blood. And so here's a vesicle that's actually fused with the plasma membrane and is now releasing its insulin. So that's regulated secretion can occur. It's also similar to what happens at the synapse, where those neurotransmitters are not kind of constantly being released. That would be a disaster for our nervous system, right? They have to get triggered by that action potential. It has to reach that threshold in order for those vesicles that are holding those neurotransmitters to be triggered in a regulated way to release their contents across the synapse. Okay, any questions about that? I think we have